Presentation today is on observed antibiotic degradation in biological wastewater treatment plants. So before I kick off, just to kind of give you an idea of how this is really relevant, is that antibiotics are not fully processed by the human body, and therefore they come out in urine and feces, which then go into the sewer, which end up in the wastewater treatment plants. Now, because wastewater treatment plants are not designed to remove these compounds, the removal efficiency can be as low as 3%. However, in some instances, we have observed that it's incredibly high, up to 99.8%. But, the kind of research, the literature shows that this variation is not particularly promising, as they're finding concentrations of antibiotics both in the liquid and solid outputs from wastewater, as well as in surface waters. So my project in particular is looking at a type of wastewater treatment that's fairly novel, but also very exciting in that it's an anaerobic wastewater treatment type, which has been growing in popularity because it offers the opportunity to recover energy from biogas. And this particular treatment type that I'm looking at, the anaerobic fluidized bed membrane bioreactor, it has very good chemical and biological oxygen demand removal rates, which, again, for those not particularly versed in wastewater treatment, that's how we measure pollution levels in the wastewater. So this is incredibly good. Biogas production sufficient to reach net energy production. So meaning that, we can actually give back to the grid from using this treatment type. And 75% removal of over 20 different pharmaceuticals, including, 3 of the biggest antibiotic categories: sulfonamides, macrolides, and quinolones. So for the purposes of my project, we've decided to focus on sulfonamides. Now, the other interesting component of this, and one of the main areas of research in my project, is this hypothesized direct interspecies electron transfer, which is a type of bacterial metabolism in which, as it says in the quote, a syntrophic metabolism in which free electrons flow from one cell to another without being shuttled by reduced molecules. And this essentially spares the bacteria the effort of having to either reduce molecules or produce external organs to share electrons with each other. And our hypothesis is that since, very effective pharmaceutical removal rates have been observed in this reactor type, our hypothesis is that this additional metabolic phenomenon might be supporting that. As it is at the moment, we've got part of my team looking at this metabolic process, whereas I myself am looking at sulfonamide degradation. So the literature, again, quite varied. This is a growing area of research, but there have been a few consistent observations across multiple different types of wastewater treatment, and that is that sulfonamides are typically better degraded when there's like another source of food. So when we test this on bacterial cultures with sulfonamides by themselves, yes, the bacteria are able to degrade it, but not as well as if they are in real wastewater where there is essentially a loss of bacterial food. So we think that that might be to do with higher growth rates, when they have something easier for them to eat. Additionally, and this is one of the critical factors in this line of research, is that when bacterial cultures are sterilized and tested with antibiotic spiked wastewater, we find that the rates of antibiotic removal drop to minimal levels. And what this tells us is that it's not a case that the bacteria are absorbing the antibiotics, dying, and then kind of sinking to the bottom of the tank, and therefore the antibiotic has been removed. It is genuine biological degradation. These bacteria are metabolizing the antibiotics, which is, of course, the really exciting part of this project. In terms of sulfonamides, again, specifically, we've noticed 3 clear metabolic degradation pathways. Again, these are scattered over different types of wastewater treatment, but we've noticed breakage in the middle of the molecule, as well as attempts by the bacteria to recover some of the nitrogen, critical to kind of bacterial growth. So they tend to take it a lot. and as well, ring break of the isoxazole ring, which again is quite unusual. Isoxazoles are typically, bordering on the toxic, depending on what's in the rest of the molecule with it. So in terms of my research projects, what I'm going to do is I'm going to set up one half of this reactor, so the half without the membrane, so then we eliminate the, not to say the false positive, but the result of having that physical separation, which isn't really what we're interested in. We will then do performance testing on this reactor so we can see how well it's performing in terms of the things I mentioned above, the chemical and biological oxygen demand removal rates, as well as sulfonamide removal rates. Metabolite detection, so we can see which pathway our bacteria are following. And then alongside all of that, we'll be doing genetic analysis to see if we can observe what changes are being made as we increase the sulfonamide dosage. And therefore, hopefully we will be able to discover which genes are being spread more widely or activated in order to achieve these results that we observe. So yeah, I hope that's my time. Still fairly early on in the research, and I haven't been able to get into the lab, unfortunately, so I don't have much data of my own, but we are working with a team in Korea who have a full pilot scale of this reactor type, and we're able to share data with them to try and get some results in the meantime. Yes, I hope that was clear.